The following content is provided under a Creative Commons license. Your support will help MIT OpenCourseware continue to offer high-quality educational resources for free. To make a donation or view additional materials from hundreds of MIT courses, visit MIT OpenCourseware at ocw.mit.edu.
Okay, so today what we want to do is we want to talk about kind of two related topics. So the first is going to be this question of the evolution of virulence and how to model host-parasite interactions more broadly. All right, that's kind of modeled on chapter 11 of Martin's book. We'll focus on the first half of it for the discussions today. All right, this is in the context of when a given host can only have one strain of the parasite or virus or whatnot inside that body. Okay? The model presented in Martin's book is very similar to kind of classic models in epidemiology, which are the so-called SIR type models, where you divide up the host population into whether they are sensitive, i.e. non-infected, infected, or resistant. Okay? And then we'll kind of draw the parallels of how we get from the model that we talked about in Martin's book, or that you read about in Martin's book, to the classic SIR models. Okay? But in both of these cases, the fundamental parameter that drives these things is this R0 parameter. It tells us about the expected number of individuals that will be, of new cases that will result when you introduce one infected member into the population.
Sorry, were you talking about super?
Only a little bit, but I would say, depending on time, but if you're interested in the superinfection discussion more, we can talk about it after class, maybe. Right. Okay, so for the second half of the class, what we're going to do, though, is that we're going to talk about the possible evolutionary benefits of sex. Right? And in particular, we'll talk about this hypothesis, which is kind of one of the reigning hypotheses for why it might be that sex is as widespread as it is, which is this the Red Queen hypothesis, right? From Lewis Carroll's novel. It's kind of and we're going to discuss this paper that you guys read about, Running with the Red Queen, which I think is kind of a nice, has a nice discussion of this debate, and then some nice experiments looking at experimental coevolution between the C. elegans worm and its infecting parasite, which is Serratia bacterium. Okay. Right, any questions before we get going? Okay, what I want to do is start by discussing this model in Martin's book, but also there's a little bit of this philosophical question. Anytime that you have modeling, you always have to make decisions about which of the details you want to try to model and which of the details you do not want to model. And depending upon the situation, it may be that some assumptions are more or less appropriate than others. Right? Now, in the model that Martin wrote down, you can we'll try to figure out what the assumptions are here. Okay? So we have these, what you might think of as some sensitive individuals plus the infected individuals are going to interact at some rate beta. So this is how the sensitive become infected. And it results in now 2 infected individuals. Okay. Now, of course, each of these individuals will, say, have some lifespan or die at some rate. All right, so this, we're going to say this die, the sensitive or uninfected individuals die at rate U, whereas infected individuals, there's an increase in the death rate described by some virulence V. Okay. Now, the model as written, what's going to be the fate of the population? Yeah. Everyone's going to die, right? And that's even true in the apps. It's not even that the population's dying as a result of the infection, right? Because even in the absence of any infected individuals, you just have people dying, right? So you need to have some way of keeping the population going so that you can study it, perhaps. And what we're going to assume is that sensitive individuals or uninfected individuals will enter the population just at some rate k. Now, right. So in terms of the philosophical question, in the beginning of the chapter, Martin talks a bit about this question of microparasites versus macroparasites. Okay? And can somebody remind us what's the distinction and what kind of parasite might this be intended to model? Yes.
What I got from it is that microparasites are on the order of like single cellular organisms, generally things that have much shorter reproductive spans, I guess. They reproduce a lot more quickly.
That's right.
And are smaller, whereas macroparasites are like on the order of a tapeworm or something.
Mm-hmm.
Which would not necessarily reproduce a lot inside of those.
Mm-hmm. Right, and I'd say that just given this distinction between the microparasites that might be viruses and bacteria as compared to the macroparasites that are things like tapeworms and so forth, it's not obvious from that you would have two different kind of modeling frameworks. But what is the argument that is made in Martin's book, or can you think up an argument for why it is that it might be this kind of model you would want to use for microparasites? Yes.
Because we don't really care about, so mentioned something that the microparasite is present in large numbers in infected individuals, so we don't have to kind of keep track of the internal state of someone that's infected.
That's right. I'd say that, and some of it's maybe even a historical thing that there might be huge numbers of viruses, a flu virus or so in an infected individual. And in some ways, maybe the number of viruses that is in that host is not the most relevant thing. And it certainly would be much more complicated to try to keep track of that. Right, so if you can get kind of meaningful predictions by rather than keeping track of the number of viruses, say, in each host, instead you just put the hosts into different classes, sensitive and infected, for example. Later we'll talk about what happens if you have a resistant type of class, right? But the idea there is that there's maybe even also some separation of time scales, right? Because you get infected and kind of quickly you're just sick and maybe infective, but at some rate you get better, and it's not that you'll necessarily gain very much by keeping track of the precise number of viruses in the host. Okay? Of course, this is ultimately an experimental observational question of whether this sort of model provides you the insight that you're going to need to make sense of these diseases. Right?
And then it also seems that the method of transmission from macroparasites should be very.
That's right. So the mechanism of transmission, and this depends very much on the disease that you're studying, right? And the macroparasites, in many cases, they're transmitted not from direct interactions between the hosts, but through through the environment or something else. Yeah. Okay. It's also perhaps just worth pointing out that parasites are just a ubiquitous aspect of life, right? So you can kind of name an organism, and you can pretty much be guaranteed that there's going to be some notion of a parasite on that organism, and there can be multiple layers of this, right? So we certainly have many parasites. We're infected by many viruses and bacteria and other things, right? But bacteria, even though we think of them as being very small, but they're also preyed upon by these phage, right? Which is a parasite that targets specifically bacteria, right? So it's not just that it's an incidental thing, but these are really viruses that have evolved specifically to divide in bacteria, right? We didn't really talk about this very much, but one of the classic models for cooperation and cheating is based on what you could think about as some sort of parasitic subpopulation within phage. Right? So this is a classic paper by Lin Chao, where he showed that if you evolve phage in bacteria in a condition where many phage infect a given bacteria, then you can evolve what you could think of as cheater strategies or cheater phage, because these are phage that maybe can't reproduce on their own, but have shorter genomes and can kind of out-replicate the normal phage. So if both of these end up in a single bacterial cell, then these cheater phage can spread by taking advantage of, the replication machinery from the rest of the phage. Right, so this is, in some ways, you might call that some sort of DNA parasite or so, right? So there's really parasites at many, many different levels. Okay? Yes?
How should we think about this K? Is it
Yeah, you know, I would say that, yeah, the K is in some ways not a very satisfying feature of this model because it makes it feel that the model is very special, right?
But I mean, in an actual population, the rate at which people are born, like, is that the same case?
Yes. So I guess that was the example I was going to give. It's not clear that that this is, of course, it requires somebody to give birth to kids, right? So in that sense, modeling this as a constant number per unit time, which is what we're doing, this rate k, right, is a little bit funny because then what you'd really want to do is say, oh, maybe that it's these guys that give birth at some rate or so, if you really want it to be accurate. So I'd say that this is, in some ways, just a mathematical simplification so that we can get at the heart of the dynamics. And what we'll see is that in these SIR models, you don't invoke anything like this, but rather what you do is you assume that at some rate infected individuals can, well, they don't just die, but they become resistant, and then maybe later they become sensitive again, right? So you just need some way of making sure that it's not the case that everybody just always dies. Right? So in some ways, this is more mathematical convenience, and the basic conclusions end up being very robust to these sorts of things. Okay. Right, so it's, in these models, it's always good to be clear about how we kind of go from this framework to something that is more of a differential equation. And what we can do is we can think about these uninfected individuals, uninfected, i.e. the S's as compared to the infected. And we're going to have these guys be X. All right, so first of all, the way that and this is S and I, right? So the way that the X will be changing is that we're assuming that there's always some influx of individuals, which could be birth or migration or something else, right, that are just always entering. Okay? But then there's going to be two ways that X is going to decrease. Right? One is that there's just a death rate that is resulting in the absence of infection. Okay? But then also there's going to be some rate of infection, which is going to be proportional to beta. Right? So this is the simplest way that you can imagine capturing this element that the infected individuals can transmit the infection to the sensitive individuals. Okay? So it's proportional to somehow. We're modeling them as it's just kind of a well-mixed population, just like in chemical reactions, and somehow the rate of infection is proportional to the kind of frequency that they hit each other. Right? Certainly the simplest kind of model you can imagine. Okay? All right, whereas the infected individuals, well, what we're going to have is we're going to have an increased rate of death. Right? So all right, so this is a simplified way to write it. Okay. All right, so this is really that there's a minus kind of a u plus v times y. So this is just the death rate. Okay. But then any individual that leaves the sensitive class, this minus beta xy, enters the infected class. All right, so this makes a lot of sense, I think. And now the question is, can we kind of make sense of what's going on? Now, all right, so you saw in your reading what this R naught parameter was, right? And you should always remember that it's defined as this thing of if you kind of have if you introduce one infected individual into a population of sensitive individuals, what is the mean number of new infections that you get? Right, and it makes sense that the key thing is whether that R naught is kind of greater or less than 1, because if it's greater than 1, that leads to this exponential kind of explosion of the infected individuals. Doesn't mean that everyone's going to die necessarily, because, we'll get into that. But if R naught is less than 1, then you expect that infection to kind of die out. Right? So why is it that when you if R naught is greater than 1 and you introduce one infected individual, it doesn't necessarily lead to kind of wipe out of the entire population? Or maybe it does. I don't know. But this model is a little funny because you always have an individual entering, right? But Right, okay, so if it's very virulent, then the infected individuals may die quickly. And this gets into this question of so there may be some trade-offs in terms of virulence. And we'll talk more about then the evolution of virulence. But I guess the question is, there's a wide variety of classes of models, not just where the ones where you have the K entering. But the question somehow is, just because R naught is greater than 1, that doesn't mean that the entire population will necessarily become infected. Right? So, because we have this idea of an exponential growth of the infected population if R naught is greater than 1. Right? So why is it that it's not necessarily going to happen that the entire population is? Well, this question is a little bit ill-posed in this model.
Is it because like the number of sensitive individuals becomes very low at some point?
That's right. Okay, and I think this is the basic intuition, that as more and more members of the population become infected, then that could, in principle, reduce the possibilities for new individuals to be kind of susceptible. Okay. Right, and I'm using susceptible and sensitive at the same, interchangeably. So, right, and so eventually this exponential rate of kind of growth of the population can be kind of limited in some way, right? We'll maybe look at this a little bit more in this SIR model because it's more clear. Okay.
So the basic reflective rate, or not, is a rate, right?
No, it's a number. Right, it's this expected number of new infections you get when you introduce one infected individual into the population. All right, and we're
It's like period. So there's not like per unit time or.
It's a number.
Okay.
Period.
So you expect that if you have R is equal to 1, you expect that when you introduce an infected individual into a population of sensitive individuals, you will get one other infected.
That's right. So that's kind of the neutrally stable situation. If R naught's 1, then you add one infected individual and you expect to get one other one. So you have a random walk, and it'll in general it'll randomly go extinct eventually. Yeah. Yes. Right. So it depends upon a number. Yeah. And so that's what we're going to do right now is see if we can reconstruct what this R naught is equal to in this model.
Another quick question, like, what's the distribution of these? So
Yes.
it's a deterministic
Yes,
model.
exactly. Right. So this is very interesting. Right. So the question is, what is going to be the distribution of the number of infected individuals, right? And in this model, we're assuming that every infected individual is the same, right? So we should be able to. All right. I wish that we just on the wall somewhere had our 5 different standard probability distributions, right? So that we could always go back to them, right? But right. So the question is, in this model, if you introduce one infected individual in the population, how many new infected individuals you get? R naught tells you about the mean, right? But will you always get the mean? No, right? All right, so let's all think about this for 10 seconds, and we will verbally kind of yell out what we think the distribution will be of the number of new infections from a single infected individual. Okay? All right, we're verbally ready. 5. All right, everybody thinks it's Poisson. Why would it be Poisson? Okay.
I mean, if the initial population is much larger.
Right. Okay. So the idea is now we imagine we're some infected individual, and there's some rate that we are infecting others. Right. Okay. Now, if I ask you the question, how many individuals will I infect in the next 10 days? Or we could do 21 days if you guys like that. Okay. So right. So if I ask, how many do I infect in the next 10 days? Right? Now, I'm assuming that I stay alive. Okay, but let's say, assuming I stay alive, how many do I infect over the next 10 days? That's going to be distributed as, that is Poisson. Right? But the question that you're asking is a different one. You're asking, what is going to be the distribution of the total number of new infections that I cause? Right? And this is precisely the same situation that we've analyzed lots and lots of times. Right? What does it look like? Okay, yes, it's going to be a geometric distribution, but why?
Well, because you can have an infection and another infection, so there's like multiple attempts.
That's right. So what we have is, because we have an infected individual, there's two things that can happen, right? Where we have, he's going to die at some rate, and he will also have another, there's this other rate which is going to go as beta times x or so, right? Telling us about the rate of new infections. And we want to know how many times we go around this loop before we degrade or die or something, disappear from the population. Does this look at all familiar? Mm-hmm. All right, were you guys the same students that were here for the first half of the class? Yeah.
And so R0
No.
is like the Vmax. Yes. It's like the number of, except it's not a rate. It's like the
mean number per burst.
Yeah, evaluated when the population.
All right, so R0 is the mean size of protein bursts in the context of this other model, which was what was the situation? And this one's a really good one for you guys to know. It's going to be useful. Yeah, so we saw this exact model in the context of gene expression, and what was the situation that we production of proteins from a single mRNA, right? Because remember we had this thing where we had the mRNA, and we said, oh well, the mRNA is going to be degraded at some rate, but there's also it's going to be translated at some rate. So the distribution of the number of times that it's translated before it degrades is going to be geometric, because we go around this loop some number of times. Right, so this is the same thing. Okay. And the paper that I put as supplementary reading by Jamie Lloyd Smith. I always get his name and Jamie Lloyd Wright, something mixed up. But yeah, Jamie Lloyd Smith. So he was studying the dynamics of infections when you have this thing where there's intrinsic variation in, say, the infectivity of an individual, right? Because here you get this geometric distribution even though all the individuals are in principle identical. Right? Now the question is, if there's some distributions of, say, infectivities, then you'll get an even kind of broader distribution of resulting number of new infections. Right? Right. So there's this classic thing of Typhoid Mary, who was a nurse that. Okay, now I don't remember the story. Yeah.
She was a cook.
A cook. Cook, nurse.
She cooked for a lot of people.
Okay, so all right, so she was somehow resistant to typhoid, but then she was cooking for other people, and so then caused a bunch of infections. Okay? Yeah. So this would be an example of a very infective individual, right? That's beyond the assumptions in this model. Right? And as you can imagine, if you have variations in this infectivity, then what it does is for a given R0, so if you fix R0, then you have a broader distributions of infectivities. What it means is that a larger fraction of the infections will go extinct, but those that get going will kind of be explosive. Okay. If you're curious about these sorts of ideas, you should look at this optional reading paper that I put out there. Okay. All right, so I just want to be clear. This is geometric number of new infections. Distribution of new infections. Yes?
So is this just one cycle? Like one?
All right, so this is right, so we're talking about the number of infections that result when you just add one infected individual into the population.
Okay, so then
Right.
you don't think about afterwards what happens to those infected individuals if they infect?
Well, we are not yet thinking about them. Although in this case, those are also geometrically distributed. But what you expect is that the mean of those things will change because the number of susceptible or whatnot individuals, that's going to change, right? But so the mean number is going to change, but the distributions will still be geometric.
But in total, that won't be geometric anymore. Like if you're looking at the total number of infected individuals after many rounds of this.
No. So just because each of these sub-steps is geometric does not mean that you end up with a geometric distribution. Indeed, if you add up let's say that I put in 20 infected individuals into the population, and I ask what's going to be the distribution of the number of infections caused immediately from those 20, that's going to be what? Gamma and for 20, or it's going to be basically Gaussian. All right. Okay. Well, if I said 100, then definitely Gaussian. Okay. Yeah. Yeah. It's a gamma distribution that looks very much like a Gaussian in that case. Okay. All right. Okay, so we've been talking about the definition of this R naught, but of course we should figure out what it is. All right, we want to know R naught is equal to, and what I'm going to tell you is that it is there's a 1 over u plus b. Okay, but then there's some other terms, and I have unfortunately lost my notes. All right, so you guys are going to have to help me figure this out. All right, and what you're going to do is you're going to take advantage of your cards, and again, put things in the numerators and put things in the denominators corresponding to how I'm supposed to fill out this equation. All right. You can start thinking about it while I give you the options. Okay. So what? Although I guess you could recapitulate this by just putting B and C. Although maybe you need it more than once. All right. Do what you will. Do you understand the question? All right. I'm going to have to. There's going to be something else I'm going to put right here, and I want to know there are going to be some things in the numerator, some things in the denominator. Okay. I'm going to give you 30 seconds to think about it because this is it's important to be able to reason your way through this. Yeah. All right, do you need more time? I'll give you another 15 seconds. All right, let's go ahead and vote. All right, ready? 3, 2, 1. All right, I like it. We're really looking quite nice. All right, so there's a claim that it's going to be AD over B, i.e. that it's going to be, we should be writing a beta K over U. All right. Can somebody explain how they got there? Yes, please.
Well, it's going to be in this diagram these two.
Okay, yeah, this helped, right? Okay, good. Right. So perfect. So yeah, exactly. So this thing is what? What is this term here?
That's the death rate.
Right, and which means that this whole the 1 over it is it's the expected lifetime of an infected individual, right? So the definition of R naught is you put an infected individual into a population, right, of susceptibles, right? Now we want to know, okay, well, there's a lifetime, expected lifetime of this infected individual, which is given by this. Okay? And then we have to think about, okay, well, what's the rate that we're going to be infecting individuals? And that's going to be beta times x, right? But what we want to know is that is x before we add any infection, right? And without any infection, then we just have a rate of entry and then a death rate. So it's just K over U. Okay? Right. Now, the key thing in all of these epidemiological models is whether this R naught is greater or less than 1. Okay? And that's going to tell us whether the disease becomes endemic or not. Right? Whether at steady state we have, we have a population of infected individuals. Okay? So R naught greater than 1 means it's an endemic population. Okay. All right, now, from in this model, we can then ask.
That's what everyone is really wanting.
All right, so do you like A, donuts, B, cupcakes, or C, carrots? Yeah, that's quite nice. Does anybody have any notion of why somebody might have? All right, so Sam likes cupcakes. Okay, that's good to know. Yeah, they're so pretty that I actually feel bad erasing it. Okay, well, we'll go ahead. We'll leave it up there for a little bit longer so that everybody can just smile because they know that the drawing is back there. All right. Okay, so in this model, the fact that R naught is this parameter that tells us about whether there's going to be an epidemic, and then indeed whether later the disease will be endemic, right? Does that already tell us that R naught is what's maximized by selection? No. Right. What was the key thing that Martin does in the chapter in order to try to understand something about what strain is selected for? Yes?
Are you thinking of when you introduce many different parasites at different times?
Right. Okay. So yes, although the part of the chapter you're thinking about is, I think, the second half is talking about superinfection, where there's all these different types and the craziness and so forth. But the initial insight about what is going to be selected for comes from a simpler model than that. Yeah, so you don't have to think about all those many parasites and those triangles and all the craziness. Instead, there was a simpler model that, yeah.
If you have multiple parasites, the study says only that one parasite exists.
That's right. And so what he does is he just writes down kind of this model of where he now allows kind of two different parasites to be spreading in the population. And at the beginning, well, we'll write it down, and we want to be clear about there's a very important assumption that he makes. Okay, what he's going to find is that selection maximizes R naught, the basic reproductive ratio. All right, R naught, and it's going to be in this model that I'm writing down now. So there's this X dot, and things look very similar. All right, now the question is, what is the key assumption that we're making in writing down these equations? Thought?
One cannot have more than one infection.
That's right. A host cannot be infected by more than one type of strain. All right, so that's very, very important. Okay. So there's no superinfection, as they say. Okay. And depending on the disease, this could either be a better or worse assumption, right? But then what is it that is defining these two strains then? In what ways are they different?
Virulence.
Their virulence is different, right? And the virulence here is, and what is virulence again?
It's how likely it is that it's going to kill you.
That's right. It's the additional mortality that is caused by being infected by that strain. Is that the only way that they're different? No. What else is different? The infectivity, right? So the betas are also different. Okay. And right, so it's important to note that in this model, it's a very simple model, but we're allowing the strains to be different in these two ways. Okay. And I think that it's very intuitive to just say that, oh, well, the beta should, you want to have a larger beta, all other things equal, right? Because you'd like to spread, okay? But I'd say maybe it's not as obvious what happens in terms of virulence. And then, of course, if you think about these two parameters in any biological context may be coupled, in which case things are more subtle. Okay?
So how reasonable is this assumption of not more than one strain?
Yeah, I think that this depends on the disease.
Is it that you're triggering the immune response and then it's
Right. So that would be that's kind of somehow the argument that and I'd say that this can, depending on whether you're thinking about the host at the level of an organism or a cell, then this would correspond to very different worlds. So certainly in the context of viral infections in individual cells, there are various mechanisms where if one strain kind of gets in, then other strains have trouble getting in, right? Or if they do get in, they can't do anything. Right, so I think this really depends on the biology of the situation, whether this is a reasonable assumption or not. Okay. All right, so I'm not going to go through all of the math because it ends up being a little bit involved. But there's I think it's the condition for the mutual invasibility of the two strains is a little bit subtle. So I do want to talk about that a little bit. Okay. All right, so in general, in this model, there is some equilibrium. X star and Y star. And indeed, there will generally be damped oscillations to this equilibrium. Okay. So this is in the model where there's just a single strain that's described by some beta and some V. Okay. Of course, you can also think about the equilibria E1 and E2 that would result when you have, so E1 is kind of what would happen, it's the equilibrium when you have X star evaluated at, for the particular parameters of strain 1, i.e. evaluated for beta 1 and for V1, X star, and that's also Y star, right? Whereas you would have a different equilibrium, E2, so this is X star and a Y star, evaluated at beta 2 and V2. Do you understand what I'm saying? So these are the equilibria that you would have if this was the only strain that was present in the population, or if this was the only strain in the population. Okay. Now, it's not obvious that those are the equilibria that result when you have both strains present in the population, right? But that's what we kind of want to try to figure out. Right? But certainly, if you only added strain 1 and you didn't add strain 2, you would come to equilibrium E1. Okay. All right. So the question is, how can we determine that if we start out at equilibrium E1, how is it we can determine what happens if we now add an infected individual, but infected by strain 2? All right, so what we want to know is, all right, strain 2 can invade. Right. And really what we're thinking about is a situation, these are the equilibria if it's the case that R1 is greater than 1 and R2 is greater than 1. Right? Because in some ways it's clear what happens if both of the R1 and R2 are less than 1, then what happens? They both die out. Right? If one of them is greater than 1, one's less than 1, then right, it's the strain that's below 1 goes out. So the only interesting or the only non-obvious question somehow is what happens if they're both larger than 1. Right? What that means is that, for example, if you had a population of susceptible individuals and you added one strain 1, one infected strain 1, one infected by strain 2, right? Then in the deterministic kind of differential equation limit, they would both spread. Right? So they're both exponentially growing. Right? Does that already tell us that you're going to have coexistence of the two strains? No. Right? It means that they're both exponentially spreading. But who knows what's going to happen later, right? And indeed, what you can show is that only one of the two strains is going to win, and it's the strain with the higher reproductive ratio, the higher R naught, or in this case. Okay. All right. So how is it that we can determine if strain 2 can invade equilibrium 1? It's if and only if something. Does anybody remember what this condition ended up being?
Y2 dot at E1 has to be positive.
All right, y2 dot at E1 has to be positive. Right. Okay. Yes. And this ends up being equivalent, I think, to what he writes. And okay, so let's write what tell you what. So the way he wrote it is that it's y2 dot with respect to y2. But I think that this is really equivalent to what you said, though. But it's a little bit. Right, because you said, OK, it's y2 dot has to be something, right? But then y2 dot evaluated at what? And you would say, oh, evaluated at E1. But then at E1, yeah. So what is y2 dot evaluated at E1? 0, right? Because at E1, there's 0 y2. So then, of course, y2 dot is 0, right? So it's almost what you want, but not quite, right? And so this condition, which looks very weird, it's really saying that, all right, if we're at E1, so there's no infected type 2 infections, right? What we want to do is kind of add a little bit of y2, and then we want to ask what is y2 dot, right? And this derivative evaluated at E1 somehow allows you to do that, right? And do we want this to be greater than 0 or less than 0? Right, we're going to do verbal answer. Ready? 3, 2, 1. Greater, right? Because if it's saying you add a y2, you want y2 to start growing. So you want this thing to be greater than 0. And this looks really crazy, but it's actually pretty easy to do because you take the derivative of this thing with respect to y2, and you just get the thing in parentheses, right? But you evaluate it at E1. So it's beta 2, and this is x star at E1, right? Minus u minus v2, and this thing has to be greater than 0. Okay. But this is x at E1. That's u plus v1 divided by beta 1. Okay. So what we have is a beta 2, then u plus v1 divided by beta 1. And this thing has to be greater than, and we'll move this over to the other side, u plus v2. Okay? So this is not so horrible, right? There's a u plus v1 beta 1, right? I just want to make sure I write u plus v1. OK, we have to put these in the denominators now somehow. OK, so we'll divide by both of these things. So we have a 1 divided by a u plus v2, 1 divided by u plus v1. Right, so we put these things in the denominator, right? And then now the beta 1 is going to come up here. So we have a beta 1 and a beta 2. And there still is a greater than sign. Did I screw anything up yet? Maybe? No? All right. OK, now we can do something wonderful, which is we can just multiply by k divided by u, right? OK, so what does this say? R2 is greater than R1, right? And what were we trying to calculate? How did we get started on this math? Right, we wanted to ask, strain 2 can invade the equilibrium 1 if and only if. Oh, no. Oh, sorry. R2 is greater than. All right. OK. Now, that's interesting, right? So that's saying that if you start out with the endemic strain 1 and you add the strain 2 in there, it's going to be able to invade if its R2 is greater than R1. That's not obvious. right? Because R2 and R1, that was telling us a different situation. Right? That was telling us about what happens when you add those infected individuals into a population of susceptibles. Okay? But this also ends up telling us about what happens when the strains are competing against each other. Right? I think it's so simple that it feels trivial, but I'm sure that if you understood it well enough, it would be trivial, but you'd have to think about it more than I have. Okay? So I think this is surprising. Now, you can also ask the same question, which is that strain 2 can, about whether strain 1 can evade strain 2, right? And that is kind of the same thing. So it all comes down to the orderings of R2 and R1. Okay? And what you find is that strain 2, if this condition is satisfied, strain 2 will drive strain 1 out of the population. Right, so if they're the same, then you can get coexistence. But it's as Martin says, it's non-generic in the sense that it's kind of a coincidence, or it's of measure 0, the situations in which the two will have the same R parameters. Right? But of course, you can imagine, just like we talked about this neutral evolution business, things are nearly neutral and so forth, you can kind of invoke similar ideas, because you can imagine that as these two become closer and closer to each other, it's going to take longer and longer for the sort of more fit strain to outcompete the less fit strain. Okay. Right. Now, even though this is such a simple condition, and the R parameters have such a simple kind of physical kind of origin or mathematical origin, the actual expression for the R's is kind of complicated, right? So again, it's a combination of all of these different parameters, where it's the beta 1, V1, or beta 2, V2. Okay. Right. So from this, you can start to think about what will be all right. Which board do you think is least useful? No. All right. Well, I don't like this one anymore. But in particular, we want to say something about the evolution of virulence, okay, in this model and what the expectation is. Okay. So what we're going to do is I'm going to give you some different situations in terms of how the infectivity depends upon the virulence, and then you guys will get to tell me how the virulence will evolve. Okay. This is virulence V. All right. All right, so first situation is if beta as a function of the virulence, so the infectivity as a function of virulence is we'll say some beta naught, right? So it doesn't. So first the question is if the infectivity does not depend upon the virulence, right? Where will virulence go to? You have equation, various places that'll help you. All right, do you need more time? No? All right, ready? 3, 2, 1. OK, we got many A's. That's great. Right. So if it's the case that it doesn't matter how, that the parasite, regardless of how rapidly it's killing the individual, if it has the same rate of getting to other individuals, in that case, we want to make the R naught get as large as possible. So then we make V go as small as possible. Right? That's the mathematical thing you can look at. And why does this make sense?
You're killing people off and you're infecting a lot of people. But
Yeah, right. So from the standpoint of the parasite, if in this situation where the infectivity doesn't depend on the virulence, well, in that case, you don't want to kill your host because the longer that the host lives, the more other individuals you can infect. Right? And this is the basic kind of statement underlying the statement that you often hear that a well-adapted parasite does not harm its host. And I think that this is one of those things that you can write down a simple model and convince yourself should be true. You can maybe find a few case studies where it seems to be true, but then you always have to be careful about how strongly you should believe such a statement based on those kinds of evidence, right? Because it's a very simple model. It's making an assumption that is very likely not true, and actually many assumptions that are very, very likely not true. And there are counterexamples, right? So Martin talks about malaria, which humans have had for millions of years, and still cause us a lot of problems, right? And so you might imagine that, well, what would happen if the infectivity is a function of the virulence? Maybe it's just proportional to the virulence, right? And this kind of would maybe make sense because you could say, oh, well, let's imagine that the number of viruses in the host, right, that somehow the additional the virulence is just proportional to that number, and also the infectivity is proportional to that number. Right? In that case, infectivity will just scale linearly with V. Right? Does that? It's kind of another reasonable world, right? Okay. So in this situation, where does the virulence evolve to? All right. We'll think about this for 10 seconds. All right, ready? 3, 2, 1. Okay. All right, so now we actually have votes that are pretty distributed around. Okay, I'm just going to write down the expression just because I think we can do it quickly. Right. So now the R naught is going to be given by, we're going to be thinking about a 1. There's a U plus V down here. But beta, now we're going to put beta here. This is A times V, and then a K over U. Right? So if we plot this as a function of V, what does it look like? Monotonically increases. This is a Michaelis-Menten type form, right? Where the R naught is a function of V, starts out linear, and then saturates. Right? And we're trying to, oops, this is not V naught, this is R naught. Okay. Right, if we want to maximize R naught, then V goes to infinity, right? Okay. There was one other model that Martin talked about, which was the situation if the infectivity itself had a kind of Michaelis-Menten type form. Right, and this was, again, okay, we still have an A there, so it's some A, V, C plus V. All right, so this would be the situation where initially the infectivity increases with virulence, but beyond some virulence, you no longer get an increased infectivity. So for example, if it's the case that every time you sneeze on somebody, you're going to infect them, then it doesn't matter if you double the number of viruses that you have, you've still saturated the infectivity. Okay? Right. Now, all right, I'll just tell you that in this case, there ends up being some intermediate infectivity that you evolve to, which comes here. And indeed, this kind of makes sense as C goes to infinity, then you end up, wait, what do I want to? Right. Oh, yeah. So if C is very large, then it just kind of looks like this. All right. It's divided by something large. But in terms of the scaling with V, it's just linear in V. Right. And that means that the evolved virulence goes very large. Okay. Does that make sense? No? Okay. So I guess what I was saying is that if C, if you're in the region where C is somehow very large, okay, then you end up with the infectivity just being kind of proportional to the virulence, right? Because the virulence, the V here in the denominator doesn't really contribute very much, right? And if you're in this, that means that it's going to be the same kind of functional form as this, except if it's A divided by C, but it's still proportional to the virulence, and that leads to a larger and larger evolved virulence. Okay. Yep. Yes.
Is it possible, like, to say something in actual infection, what the data is virulence, or like, can you
Yeah.
go further than just these verbal?
Yeah. Right. So I think from real data, I think people do argue about it, but I think that it seems to be, I guess if you had to, there is some sense that it does plateau, but it is a function of, it's an increasing function of V, but not sublinear, right? And the question is how strong of a statement you can make there, because also in many cases there is superinfection, and superinfection tends to lead to even higher virulence than what you would expect from this, because you're competing against viruses in the same host, right? So you kind of have to outcompete the other parasite in the host as well as kind of go on. And you don't pay the full cost associated with keeping the host alive, right? So when you have superinfection, there's this notion that the better strategy from the standpoint of the parasite can be to just evolve very high virulence, right? So you're going to kill the host, but you can get out quickly, right? So then you can imagine that the less virulent strain was kind of stranded in that host that died, right? So from that standpoint, you can think about in some cases for a parasite, evolving low virulence is somehow like some cooperative behavior, because you're keeping this host alive so that you're using the resources in a wise way so that you can infect other individuals. But then that kind of strategy is susceptible to these cheating strategies that just have high virulence and kill the host and then get onto a new host, right? So I think there are enough of these sorts of issues that it's hard to take any of this too seriously. I would say that perhaps where this field has had the biggest impact is in the context of vaccinations. All right. Because you can really measure R naught for many different diseases, and I think that that's somehow easier to measure than many other things, because you can try to do tracing of infections, right? So if you think about somebody that gets infected and moves to a new city, or lands in a new city, you can try to figure out who they infected, right? So then you can go and measure these R naught parameters, right? And of course, the diseases that we worry about tend to have R naughts larger than 1, right? And how large they are tells you about how difficult the kind of vaccination will be in order to be successful and to remove the disease from the population, right? So if you're curious after class, you can come up, and there's a nice table that I have here for smallpox, measles, whooping cough, German measles, chickenpox, diphtheria, scarlet fever, mumps, and poliomyelitis. Estimates of R naughts, right? And they kind of range. They're kind of 5 to 15, to give you a sense, right? And so which diseases? Right. So if you want to remove the disease from the population, what is it that changes in terms of vaccination in order to remove the disease? Yes.
In R naught, there is a K over U as the population available.
Mm-hmm. That's right. So you by vaccinating, you're kind of removing susceptible individuals from the population and making them resistant somehow, right? And the R naught parameter tells you about what fraction of the population you have to vaccinate in order to remove the parasite from the population, right? So basically, you have to vaccinate a fraction of a percentage, or P that's greater than 1 minus 1 over R naught. Okay. So as R naught kind of gets very large, it means that you have to vaccinate essentially everybody. Okay. if you have R naught of, say, 5, which is typical of many of these diseases, it's saying you kind of have to vaccinate kind of 80% of the population. Right? You can never get to 100%, right? And that's why it's very difficult to get rid of these diseases with large R naughts. And incidentally, they note that smallpox has an R naught of kind of 3 to 5. And this always depends on the environment, but in the case where they measured, smallpox R naught is kind of 3 to 5. And this is sort of small as compared to many of these diseases. And this is telling us that smallpox is easier to get rid of via vaccinations than many of these other diseases. And indeed, we've, the vaccination procedures have been more successful in smallpox than the others, right?
Do you know what is Gore?
I don't, but if maybe in the next 20 minutes somebody can Google this, and at least we can estimate this right now, though. I mean we've had 5 Ebola patients come to the United States, and we've gotten 2 or 3 infections. So I'll say 3/5. It obviously depends on the environment, right? Yeah. Yes.
I mean, I guess that was my question. Like, you're not going to take someone with Ebola and throw him in New York City and just measure, how many people can check.
That's right.
So like,
yeah, but the thing is, you don't have to do anything that's so immoral, because what you're interested in is the R naught for an individual in an actual environment that they're actually going to be in, right? So this is a situation where, the doctor comes back from Africa after working with Doctors Without Borders, right? He knows, in this day and age, he knows that he has to watch out for a fever, and then if he gets a fever, he calls in and he's brought to the hospital, right? And that's the world that we're interested in of what the R naught is, right? It's not the world in which, there's fevers everywhere and nobody knows, right? So it's so the R naught in the United States is going to be much lower than the R naught in, somewhere else, right? Yeah.
Is there a notion of R naught for a disease that you don't transmit between people, like you get malaria or plague?
Yeah, right. So I think that you could try to generate a similar kind of R naught for those diseases, although I think that it's going to be very muddled, right? In the case of where you have all the vectors and so forth, because it's not even clear. Yeah, okay, I'm hesitant to say too much because I don't know anything.
According to Wikipedia, it's like 1.5 to 2.5.
For Ebola?
Yeah.
Here or in?
It says the 2014 West Africa outbreak.
Okay, so this is in West Africa then. Okay.
Apparently.
Right. Yeah, and the idea is that it has obviously spread exponentially, which means it had to have been larger than 1. Right. And I think it's important to remember that just because you have a chart with a bunch of R naughts, this is not set in stone, right? Public policy, hygiene, and everything changes this, right? And we'd like to drive it down. Yeah. Was there a question in the back?
I was just going to say it's about
Same thing, about 2. Okay, all right. Right. So that means that that we can actually, in principle, vaccinate against Ebola. We only have to get, over 50% of the population vaccinated in West Africa, and then we can maybe make it so it cannot spread and become an epidemic, right? Of course, we need to have a vaccine first, right? All right, so I think that I'm going to skip the discussion of SIR models because you are going to play with some of them in the context of your problem set. And if you just Google SIR, you can find it. Very similar kind of intuition that you get from this model, okay? Because I do want to spend the last 15 minutes at least talking about the evolution of sex, because it is an interesting topic, and I think that the paper is kind of a nice discussion of it. All right, so can somebody say why it is that this is kind of a puzzle at all? That's right. All right. So sex is costly, and in particular, if you have this obligate biparental sex, right? Then there's, in particular, there's the so-called twofold cost cost of males. Because you can imagine comparing kind of these two populations, right? One of which has both males and females, and one of them is just maybe either reproducing asexually or parthenogenically or hermaphroditically or whatnot. Yeah. Okay. And you know, so you can just imagine, well, if you have a male and a female, then on average, if they have 2 kids, you end up with another male and a female, right? And you know, of course, this can be many different males and females, so you don't have to have any sibling anything, right? Okay, but if everybody's having if every generation, each female is giving birth to two progeny, then you end up with a constant population size, right? Whereas if you started out in a population with just 2 females and they were reproducing hermaphroditically, then you end up with, whatever, more, okay, 8. Okay, so you can see that you get a factor of 2 in the rate of exponential growth of the population. Yeah.
But I mean, it seems to me the question is not why sex, but like why males. See what I mean?
Yeah. Yes. Yeah, okay. Right. So I think that. Right. So this is the most extreme cost of sex, right? But then also you can think about even just horizontal gene transfer among bacteria, that it's a costly behavior in some way or another. It's not as costly as this, right? But if you're going to think about bacteria that in their competent state, when B. subtilis kind of pulls in DNA, it stops dividing, and then it enters a state where it kind of reels things in, and it can pick up DNA that may be harmful in some cases, right? So there are various costs that are for the, and if you want to call it horizontal gene transfer sex, right? But right, so the key thing of sexual reproduction is that it somehow is the sharing of the DNA, and I'd say that this can either be relatively low cost or relatively high cost, but this is the most extreme version of it. And I'd say as a species that reproduces sexually with obligate biparental sex, then I'd say that, and not only humans but almost all animals have this form of reproduction. I would say it's sort of surprising, given that this is a huge cost. Right? Yeah. So but it's not that all species that engage in any sort of gene transfer bear this large cost, but they bear more modest costs.
Although,
Yeah.
of course, a bunch of species do, deal with this by just having multiple children per sex.
You're just saying that they can just have more kids.
Yeah.
Yeah, although, I mean, the basic statement is still true, though. Let's say that these females all have 3 kids. And they get to grow exponentially, but then these guys grow faster, and ultimately there's going to be competition for resources, and these ones will still win.
Yeah.
Right? So the question is, if you can imagine you have a population that's reproducing sexually. If one female has a mutation that leads her to start reproducing parthenogenically, that mutation you'd expect should spread throughout the population very rapidly. Right? And indeed, there are these cases of, for example, sharks held in captivity, where a female held in captivity for years eventually gives birth to daughters. Right? So this, and as far as so this sort of virgin birth is possible. I'm not aware. Okay, well, I'm not going to talk. Okay, but yeah. So, but in some animals, it is at least possible. I'll say. Okay. All right. Okay. So then you have to ask, well, okay, well, what kind of selected advantage could sexual reproduction have that could possibly compensate for this so-called twofold cost of sex, or twofold cost of males? Okay. And what is the argument that's made in this paper? Anybody? Yes?
Recombination favors genetic diversity.
That's right. Right. So this recombination is favoring genetic diversity. All right. So there are a number of different mechanisms. I'd say that at the heart of it, okay. So this idea of the red queen hypothesis is, all right. So let me see if I can find the actual quote for you guys. Maybe not. So it's from a Lewis Carroll novel, Through the Looking Glass. The quote was something, you have to run as, oh shoot. All right, never mind. Okay, I can't remember what the quote was. The idea is that sexual reproduction may allow a population to adapt against, say, changing environments more rapidly. Okay? And this has a couple of reasons, because you generate genetic diversity. You don't have the same clonal interference kinds of effects that we talked about earlier. All right? So if you have asexual lineages, then if you have two beneficial mutations, they cannot both fix. Right? So the more fit version is going to fix, and then you have to wait for the next one. Right? Whereas in sexually reproducing populations, those genes can spread throughout the population sort of as genes rather than being tied to a particular individual. Okay? So that means that if you find yourself in a new environment, it may be the case that sexual reproduction can allow for the population to adapt more rapidly. But on one hand, you might say, well, the environment's always changing, so that can always favor the sexually reproducing populations. But then there's a feeling out there that maybe that's not enough, in the sense that the environment's not changing rapidly enough and dramatically enough to force the population to reproduce sexually as compared to asexually. Right? And so the proposal from the Red Queen hypothesis is that the constantly changing environment is a result of coevolution between hosts and their parasites. Right? Because parasites are always trying to target kind of common host genotypes, because they can spread on those. Right? So the parasites are evolving, but then the host is sort of host population or genotypes are kind of being chased away by those parasites that are targeting them. Right? So the notion is that parasites, as a result of this coevolution, can be the source for the constantly changing environment that may be driving the evolution of sex. Yeah.
I guess the question I would have is, bacteria also get parasites, as you pointed out.
That's right. Yes, right. So the question is why, if...
Why only in multiple?
Yeah, right. Okay, so I can give you my best guess on this. All right, first of all, I would say not everybody agrees that the Red Queen kind of hypothesis is the sort of true explanation, if a true explanation even exists, right? But within this framework, you definitely want to try to explain why it is that kind of large life forms seem to have a lot of obligate sexual reproduction, right? Because this is a very strong pattern that you see, right? And I guess what I would say is that there's certainly a correlation between size, physical size, and generation time, right? And that generation time tells you something about the typical timescales over which you can evolve, right? So my sense of this is it just may be that that animals, in general, large animals, they by their nature will evolve rather slowly as compared to their parasites, and so that means that they are kind of the populations that are most in need of evolving, of speeding up their evolution. And it's hard to know how convincing that argument should be, but yeah. Yes?
What's the difference in reproductive time between phage and bacteria? Like how fast is it?
Yeah. Right. Okay. So bacteria can, as we've discussed, division times are kind of order hour, and phage, I think, so when you get a phage infection, so what happens is that the phage, you can infect as a single phage, and then they will generate burst, they'll kind of divide within the bacterial cell, and then burst out as a population of 100, 200 kind of typical phage. And I think that might take 4 or 5 hours, is kind of my sense. All right, so I'd say that, and then they go off and they find new bacteria. So in that sense, they may have, so it's a little bit faster than the bacteria, I'd say. Yeah. Okay. All right, so what, can somebody say what the core experiment was that they did in this experiment? Yes. That's right. So there are going to be, so this worm, C. elegans. It's sort of a millimeter in size, right? And there are going to be 3 different conditions. One is there's kind of the wild type that can outcross, can have males mate. Some that are then there's the obligate outcrossing, which means they have to mate with males. And then there's the obligate, what do they call it? Obligate selfing? Obligate selfing, yeah. Okay. And then what do they do with those worms? That's right. So then there's a bacterial pathogen, Serratia marcescens, that, and they're going to have these 3 different conditions, right? Where they're going to, for the SM, the bacteria, they're either going to have, they're going to allow for coevolution, where they take the bacteria from each of the infections and propagate, or they're going to do kind of the no evolution, where you just compete against the ancestor, right? And there's also control. Okay, so there's coevolution, there's kind of this no evolution of the bacteria, and then there's also control where there's no bacteria, right? And what was the most striking thing that they saw in this experiment? That's right. Right. So if you allowed the bacteria to be evolving against this obligately selfing population, this killed the worms. Right? And what was the other thing that was maybe very striking about their experiment? Yeah.
The worms that could either outcross or self.
Yep. Yeah, so this was kind of amazing, right? So they saw, as a function of time, if you look at outcrossing, so kind of the rate of mating with the males, this started out at kind of like 0.2, and then in the presence of the coevolution, it kind of went up, and it goes up to 80, maybe 80%. Okay, and for coevolution, it stayed up high, whereas if the wild type worms, if they continued to be just challenged by the ancestral bacteria, it initially came up, but then it came back down. So this is the coevolution, and this is the ancestral SM. Right. So there was a sense that wild type population, it initially it evolved to outcross more, but then once it had solved this problem of how to handle the ancestral Serratia, the outcrossing rate went back down. Right. So we are pretty much out of time, but I do want to. I don't know if you guys noticed the last sentence of the paper. It is amazing that they got this through the publication process, right? All right. So so they say, taken together, the results demonstrate that sex can facilitate adaptation to novel environments, but the long-term maintenance of sex requires that the novelty does not wear off. It's one of those things that you read and you think. Okay, so I will leave that sentence with you, and then we'll meet on Tuesday. Okay.